Yeah, I'm going to present AlphaGenome, which is really came out last week on the Nature. Um, you probably know this is a model that following up the work on Informer and Borzoi, basically the same series of model to predict regulatory variant effects. So before we dive into the model, just give a very brief overview what we're doing at Google DeepMind, the science team. So we publish like science models working on very diverse different field, and biology is actually one of the focus. We have obviously AlphaFold, and as Julia mentioned, we have AlphaMesons focused on Mesons variant effect prediction, and some other biology models and non-biology models. Yeah, that's kind of the focus what we do. And in terms of genomics, we're working on gene expression prediction for a long time. If you look at the gene expression regulation, it's actually very, very complex process. It has transcriptional regulation and post-transcriptional regulation. Transcriptional regulation, we're talking about enhancer promoters and DNA accessibility, transcription and translation, and RNA splicing. We try to cover as much as we can with AlphaGenome. AlphaGenome really builds on top of many, many years of research from this field. If you look at previous models, Informa and Borzoi, they're working really, really well. They take very long input lengths, and the limitation is that they are typically a bit lower resolution. Some other type of model, they have very high resolution up to 1 base pair, but then they have it takes relatively shorter sequence as input, therefore has less context. And there are some other models that specialize on single modality, and here we try to cover as many modality as possible. And with AlphaGenome, we really try to advance on two sides. But on the resolution side, we push the model to 1 base pair resolution and try to cover gene expression, splicing, accessibility, many different things, and we take 1 million base pair as input. The model is kind of a standard unit model with some architecture innovations. So we have a sequence encoder, which is basically convolution layers, and a transformer tower, which let the sequence talk with each other, and in this way we increase the effectivity, the receptive field of the model. And in the end, we have a decoder which brings back the resolution to 1 base pair to 1 base pair. And in the end, from the embedding, we predict the different modalities. This varies from RNA-seq coverage, cage, and in image. Like, actually, one thing to note that the previous model, the previous version, which is Informer, is actually more expensive to train than AlphaGenome. It's actually Informer requires 50% of this model actually takes only 50% of the training compute compared with Informer, which is really nice to know, because then we can experiment much faster. Another thing to note about AlphaGenome is this distillation. Distillation is kind of the idea has been there for a long time. The thing that the specific way we did the distillation is we train multiple fold, like we train multiple models, and in the end we try to ensemble the model. But then ensemble means that you need to inference each model, and with 6 models you need to run 6 inference paths, which is quite expensive to run. So we're thinking like how we can combine these models together with one single inference run, but with the same performance, maybe even better. This is where distillation comes. And the basic idea is that after you train this pre-training this teacher model, you try to use a single model with same architecture to distill what the knowledge from the teachers to the students, right? One thing to note that if we introduce some mutation in the input sequence so that there's a lot of sequence diversity during the distillation process, and the student model is initialized randomly, and the training data all coming from the teacher, let's see. And that works pretty well, in fact. This is after training the model, we look at a lot of visualization. This is one of the example. We look at 1 million base pair region. Actually, if you look at the 1 million base pair region, one thing to note that there are typically many, many genes, right? In this case, there are quite a few genes in this region. We take a very specific cell line, and this plot always shows the positive strand RNA-seq, negative strand RNA-seq, and always has observed one compared with the predicted one. Yeah, one thing to note is the we always predict the positive and negative strand simultaneously. And within this, you see that the RNA-seq coverage actually predicts pretty well. And with other tracks as well, like the peaks generally predict pretty well. If you look at zoom in a bit into the detail, this is one specific gene that within this region, right? And again, the splice size are pretty well, which is pretty quite important to, because with one base pair resolution, we can predict precisely where the splice size are, and also the splice site usage. This is a number between 0 and 1, indicating the percentage of the splice site being used by all transcripts. On the another thing that we innovated in this paper is a splice junction prediction. This is to tell like what all the different intron possibilities are there, and as well as the expression level of the intron, basically the strength of the intron. And so here is just to show some more examples. These are different genes we're showing. One thing to note is the model predicts multiple tissues and cell lines, and sometimes the tissue specificity is captured very well. Like this gene, I think is predicted pretty well, and is expressed in these two tissues or cell lines. Some other cases, like in this case, the tissue specificity is not that great. In the observed, we see there's quite a bit of expression in this liver tissue, but its prediction is pretty weak. The magnitude is much smaller. So this is just to say that sometimes the tissue specificity works well, sometimes not, and the magnitude can be very wrong. On the resolution side, the exon coverage, I think the exon is predicted pretty well, aligns well with the observation, and with borzoi, which is a lower resolution, you can't really predict the exon boundaries precisely. We evaluated holistically on the track prediction. So track is basically all the, for instance, RNA-seq is one track, one strand is one track, attack is another track, and each tissue is one track. So track prediction performance is basically means that how well you predict the holdout training data. This is evaluated on holdout intervals. And we always compare with the best model out there, even though we compare with every model, but in this plot we only show the best other model. And showing one example on the gene expression prediction. I think both the cross gene, which is like per track prediction, and also the cross track, which is tissue specificity. Both of these we compare with borzoi, and we think there's quite some improvement. This is one example. This goes to other modalities as well. On the variant effect prediction, we see also universally the model is actually improving quite a lot on many diverse variant effect prediction tasks. Yeah, this is a really large collection of variant effect tasks. I think it really takes takes a team like a lot of effort to curate all these different evaluations. The on the eQTL, just show one example on the eQTL effect size prediction and causality prediction. We compare with some other models like Bosoi and ChromaBPNet. We generally see quite big improvement on effect size and causality. And on the cEQTL, this one is. And one thing like we see constantly a bit of challenging is distal variant effect. So, for instance, on the eQTL sign prediction stratified by the distance, the variance to TSS, and we see that with the distance increase, the task become more challenging for all models. Our model is better, but not is still very far. Another task we look at is encode our E2G task. Is linking like enhancer to gene. The model is a bit better, but I would say like this is still quite challenging task. So another thing we did quite a bit on the model is really the RNA splicing part. So if you look at previous splicing models, actually this is other models are pretty strong on this task, like especially Splice AI is really widely used in this field. And Bosoi, they do RNA-seq and prediction and splice set, kind of indirect. And some other models do splice site and or splice site usage prediction. I think our model predicts all the different relevant quantities for splicing, including the RNA-seq coverage, splice site and their usage, and also the junctions. This is just to give you some examples of the splicing prediction and in terms of their predicting variant effect. For instance, in this case, is a deletion. This small deletion create exon skipping, and this exon skipping is clearly predicted. If you look at the splice junction prediction, we predict this this new junction being formed, and the previous junctions are managed. And if you look at the coverage prediction, the RNA-seq coverage, we see like generally lower coverage on the skipped exon, which is consistent with splice site and junction prediction. Some other examples where, for instance, in this case, the mutation created a new splice site, basically you see that in this case, the exon being elongated with a new splice site. Yeah, and another thing we double checked is, with in silico mutagenesis, we want to check if the model has learned like, the sequence features that are relevant for RNA splicing. We took a very specific example. In this case, the splicing motif being highlighted as well as the poly T track and branch point internal motifs as well. This just to show that the model actually consider the splicing related motifs when it makes these kind of predictions. We benchmark quite extensively in terms of RNA splicing on different variant effect prediction tasks. It's coming from SQTL, like splicing QTL, and stratified by distance. And in general, we see some improvement. And another thing is this is a very challenging task, predicting splicing outliers. It's a GTEx splicing outlier. The task is really distinguishing rare variants and say which of the rare variants is causing aberrant splicing. we see quite some improvement on the model. On the ClinVar task, this is really predicting which of the variant is actually annotated as pathogenic and which one annotated as benign in ClinVar. This is a binary classification. We stratified the task by different regions, and we see cross-board the model is quite strong. And on the MPRA, we see that the model is strong, but slightly worse than pangolin, this other model. I think naturally the model is trained on very, very long context, and this MPRA setting, the input sequence length is actually very, very small, and we see that in many cases. So the model is generally sometimes can struggle on MPRA setting because of the input sequence length. Yeah, the another quite interesting thing about splicing is deep intronic cryptic splicing. The this is actually quite challenging to predict. We looked at a few examples. In this case, for instance, this variant caused kind of a deep intronic cryptic splicing and create a cryptic exon here. And the model predict precisely there's some exon skipping on that cryptic exon, but we see that two new junctions being formed, supporting this cryptic exon. And with this model, the core difference between AlphaGenome and maybe Splice AI is that Splice AI predicts splice size, but we predict more than that. We have a really holistic overview of the molecular consequence of that variant. And we can do in silico mutagenesis and see what kind of motifs it captured. Obviously, in this case, it creates a splice site. Naturally, you see a splice site motif here. This is another example of deep intronic splicing, which is kind of similar. Another thing, because we predict RNA-seq coverage, one thing that we captured, which not necessarily obvious, is the poly-A signal. Because we predict the coverage of the UTR as well. So in this case, a variant is on the poly-A motif. And you see that there's quite a bit difference between the poly-A side. If you compare the red and the blue line, this basically indicating the variant and the wild type. We see that there's elongation on the poly-A side with the variant. And we actually benchmark the polyadenation QTL and stratify this by distance, and we see some improvement over borzoi. Yeah. So as mentioned before, that one thing we really try to build is this multimodality, and we think this is quite important. Not only that, it gives you some overview on the variant effect across multimodality, but also hopefully these kind of different modality they interact with each other and gives you some better overview of the variant. And this is one example. We try to show the power of multimodality. This is some variants from tau1 gene, and those variants, and this gene actually associated with leukemia. We know from literature that some rare variants from this variant actually associated with this cancer. And we analyze some known oncogenic variants, both single nucleotide variants and also small indels. So you probably know that we support these indels natively, and it's actually quite a lot of work to make indel scoring work. But so in this case, we look at these three cluster of variants. One cluster is above, and this is a negative strand gene. One cluster is above the gene, upstream of the gene. One is close to the TSS, and another is downstream. And these are kind of known mechanism that these variants cause cancer. And by regulating a tau1 gene expression, actually upregulate that gene expression. So if you look at the those variants in general, what a pattern you generally see is that if you look at across RNA-seq and histone modification and DNAs, you see the histone marks which associated being activating that gene is being upregulated. So each of these track is a difference predicted between is difference of the alternative versus reference. And the positive numbers, this means the alternative is higher. And in this case, the histone modification is being activated, and these marks corresponding to activating the gene, right? And the other marks representing repressive histone marks, we see that is kind of downregulated, and all of these collectively leads to upregulating of this oncogene expression. As a control, we also look at we random sample some variants in those different buckets, stratified by length of the variant. We have SNPs and also different lengths of indels. And what we generally see is those oncogenic mutations, they are generally much higher. They has much higher predictive effect compared with some random sample gene variants. And in terms of mechanistically, when you do in silico mutagenesis, you generally see that we recapture this non-MYB motif, and this is some motif that previously non-invalidated. Another thing, one of the major reasons that we're working on this AlphaGenome is really we try to resolve more VUS, and obviously this is quite a big problem in the field. With the model being more accurate, hopefully we can help to resolve more VUS. And taking some examples from this paper, actually we updated this in the supplementary notes in the published paper. This is not in the preprint. We did some very extensive analysis for the variants presented collected in this paper. They vary by mechanism, right? Some of the promoter enhancers, some others UTR, both 3' UTR, 5' UTR, and the mechanism can be very different. It's either mRNA binding or some other. There's obviously a splicing variant. So this is a kind of the holistic view of different non-coding variants, and they're collecting from different mechanisms. And we evaluated our model on the variants suggested here. Some of them are successful, some of them are not. One successful example that we presented, and we think that we can actually maybe help resolve this VUS variant, is a variant in the 3' UTR. And this variant actually is having an effect on the polyadenation motif. There are some other variants in ClinVar in this same motif, actually annotated as pathogenic. Through our model, we understand that this variant actually contributing or affecting the same polyadenation motif, and they have the same mechanism. And in this case, there's some quite strong effect predicted by the model. And we see with the alternative, with the variant, the gene is generally downregulated. And we also did some quite comprehensive ablation studies. And the ablation study are generally very, very interesting. I encourage you to take a look. We trained a lot of different versions of the model, varying by resolution, the sequence length, and comparing ensemble models versus distillation, and also if multimodality is going to help. So with the resolution, because one claim we are doing is that we train the model as one base pair resolution for some of the tasks like RNA splicing. It makes sense to be really, really precise at base pair resolution. Some other tasks, maybe you can afford to be lower resolution, like expression, if you're going to aggregate expression on the exon region. This is ablation basically to tell you that in general, what we see is we present like track performance and also variant performance. So there's some distinction between track performance and variant performance. In this case, when you vary the target resolution, the track performance generally goes a bit down when you decrease the resolution, and the variant performance it varies by task. Some of the tasks like SQTL tasks, the performance goes down. Some other tasks may be more stable when you increase the resolution. Yeah. On the sequence length ablation study, we present like there's different ways you can do that. One way is you train a 1 million base pair resolution, and you at inference time you inference that model at different lengths. So you can actually take 1 million base pair 1 million base pair model and inference, let's say, on 500,000 base pair. And this is what we're doing. This is the blue line. And what you can also do is train a smaller input sequence length model and apply that at inference time to longer sequence. And this is the purple line, and the green line is basically train on shorter sequence lengths and evaluate on shorter sequence lengths. So one interesting comparison is really, can you train a 1 million base pair model and apply that to different sequence lengths? Is that the same as train model on that length and inference on that length, right? This is what we show. In general, the two ways of training models are very, very similar. So you can actually, this is what exactly we are doing. We train a 1 million base pair model, and we can deploy time, we can allow the model to inference at different sequence lengths, and we see that this generally works roughly the same if you train and evaluate at that length. Another thing, as mentioned before, is distillation. So as mentioned, it's quite expensive to run ensemble, let's say 6 different models. And we want to compare that, what's the gap between distilled model and ensemble model, right? And for most of the cases, it's quite nice that we see that the ensemble model is not that much better than the distilled model. And there's certain cases where like the distilled model is actually slightly better than the ensemble model. We think this is because the distilled model see much more sequence diversity. Yeah, and all of that obviously reduce the inference cost. Another thing is multimodality. We have some major modality, and the experiment we are doing here is really, for instance, if you just train on RNA-seq or on expression modality and evaluate the model on some other on all the tasks, obviously, if there's a task synergy basically between these different tasks, right? So one thing to note is accessibility and contact map. It turns out that for accessibility tasks like CIQTL or contact map, it is actually the same. There's not much task synergy. You can train a model only to do a task-seq on DNAs, and it works almost the same, versus a model that does everything. There are some other tasks like eQTL, which is basically corresponding to expression modality. And if you look at here, the green bar is actually lower than the dashed line. The dashed line is the full model, right? The green line, the green dots are basically the model that trained on that task. only and evaluate on that task, you see the gap based in indicating how much other modalities actually contributing to this task that they're interested in. There's quite a bit gap on the eQTL and some other expression related task. So in terms of model release, we released the model API, I believe like 6 months ago or more than that. The API is pretty nice to use. Hopefully, you agree. Like after using the API, each of the inference tasks, for instance, one variant takes only less than one second. You can get the prediction for all the different modalities, and we have also plotting library to plot time like that. Obviously, this is a moment for non-commercial use. We also release the model weights, and you can do some fine tuning, for instance, with the model weights. There are quite some limitations with the model. Regulatory variant effect prediction, there's so much to do still. And for instance, the distal enhancer is still quite challenging. The cell type or tissue specificity, we improved a bit, but I think there's still much to be done. Obviously, the model is trained on a fixed number of cell types and tissues, and with this multitask setting, and it's pretty hard to generalize to some other new cell type or tissues. Maybe you can do fine tuning on that. The model is only on human and mouse. There's I find it's pretty hard to improve on the variant effect, but maybe with more data, this is possible. To summarize that, we present a quite comprehensive and accurate DNA sequence model for human regulatory variant effect prediction. If you think like the model like published in the team, previously we do alpha missense on the missense region, and with AlphaGenome we do non-coding variants. Hopefully this represent a more comprehensive solution on variant effect prediction, and the long DNA sequence context and high resolution. And I think this is a pretty nice way of solving that. There's a quite extensive of engineering went to how we do variant scoring and model distillation, and this is done now running pretty fast. And I think this is a very nice platform to extend to any new data set or assays or even more species. Yeah, with that, I'd like to thank the team. It's really a huge teamwork with all the people on the picture. And thank you for your attention. I can take some questions now.